the glomerular filtration is highly due to the hydrostatic pressure that's created by that difference in diameter between the afferent and efferent arteriole. So, and we try and keep that glomerular pressure, that hydrostatic pressure pretty constant because we like a consistent rate of glomerular filtration. And then that filtrate is captured in the capsule and it runs through this proximal convoluted tubule, which has a very high reabsorption surface area due to microvilli. It's going to, assuming that we're within physiological levels, then 100% of amino acids and glucose will be reabsorbed, and about 65 to 75% of the sodium and the water will be reabsorbed. So today we're going to look at the loop of Henle, the distal convoluted tubule a little bit, and then also the collecting duct. So we're going to go through the rest of this tubule system and look at how the remainder of water and sodium are reabsorbed, as well as how the kidney produces either a dilute urine or a concentrated urine. Hydrogen ions can also be secreted. Bicarbonate can be reabsorbed or secreted depending on acid-base balance. We're going to leave hydrogen ion secretion and then bicarb secretion reabsorption for next week when we're going over fluid balance and acid base. So for chapter 24, we're going to focus on things like glucose, amino acids. So how much is reabsorbed, where is it reabsorbed, as well as water and salt. So those are kind of the four big ones. We'll touch on urea. Urea is a nitrogenous waste product, and we're not going to go into what's called the urea cycle. We're just going to talk about how urea contributes to this concentration gradient. So just a tiny bit. So one main function of the kidneys is to maintain body fluid osmotic concentration at around 300 milliosmol. Osmolality is the number of solute particles in 1 kilogram of water. So if you go back to 1141 and talking about osmolality and osmolarity, then glucose would contribute 1 particle because it doesn't break apart in solution, whereas sodium chloride, because it breaks apart into sodium and chloride, contributes more to osmolarity, osmolality. I'm just going to mention now that osmolality and osmolarity are used interchangeably. The difference is zero, essentially. Osmolality is the number of solute particles in 1 kilogram of water. Osmolarity is the number of solute particles in 1 liter of water. But guess what? 1 liter of water weighs 1 kilogram. So assuming water is at 4 degrees, which it isn't in our body, but close enough, as far as when we're talking about values, these terms are going to be used interchangeably. They'll be used interchangeably as I'm talking and in your book and in diagrams. So for our purposes, there's no difference between osmolality and osmolarity. It's the number of solute particles dissolved in our solution, which is plasma. Okay. The way that this osmotic balance is maintained is through what are called countercurrent mechanisms. There are 2 types of countercurrent mechanisms that are used in the kidneys. The first is called the countercurrent multiplier. It creates an osmotic gradient in the medulla of the kidney. The second is accomplished by the vasa recta. So this countercurrent exchanger is the vasa recta, how it maintains this gradient, but it still reabsorbs water and salt from the nephron loop. Okay, so two types of countercurrent mechanisms. We're going to start with the countercurrent multiplier. It is created by the interaction of filtrate flow, meaning that in the descending limb, filtrate is flowing from the cortex to the medulla, and then in the ascending limb, it's running from the medulla back up to the cortex. So countercurrent means that in this hairpin loop, we have flow that is in different directions. So it's going down and then it's going back up. So countercurrent. The other factor that's going to be really important is the difference in permeability. So there are two things that are required to make this setup work. The first is that the limbs run countercurrent. The second is that they have a difference in permeability. Then we'll talk about the countercurrent exchanger, where we have blood flow that is also runs in different directions in the vasa recta. These two countercurrent mechanisms work together to establish, that's the countercurrent multiplier, and maintain is the countercurrent exchanger, a medullary osmotic gradient. This medullary osmotic gradient is then used by the collecting ducts to determine if urine is going to be dilute or concentrated. So the loop sets it up, the vasa recta maintains it, and then the collecting ducts are going to use it. So we keep talking about this gradient. You're like, what is this gradient? What does that mean? Just let's say we're just talking, let's say 100% of the solid molecules were salt, which they aren't, but for simplicity's sake, let's say everything is salt that's dissolved in the filtrate. Up in the cortex, where filtrate is formed, the salt concentration would be 300 millimolar. And then that creates an osmolarity of 300 milliosmoles. As the filtrate progresses down, the interstitial fluid, okay, so not the filtrate, but the interstitial fluid of the medulla, of these medullary pyramids, is going to increase from 3 to 4 to 5 to 6, all the way down to 1200 milliosmoles. So the salt concentration, as we progress down from the cortex into the medulla, increases. So once you get to the papilla, the amount of salt in the interstitial fluid is going to be much, much higher. So 4 times higher, that multiplied. It just keeps getting more concentrated and more concentrated and more concentrated until the concentration in the interstitial fluid is very, very high. This is showing that there are the 3 players. These are juxtamedullary nephrons that are going to be associated with this countercurrent mechanism. The cortical nephrons, their loops barely reach here, so they don't come into play. This is going to be, excuse me, juxtamedullary nephrons that are creating this gradient. And then the vasa recta is a series of capillaries that overlies the loop of Henle. They are going to also be in play, and then the collecting ducts. So this is just reminding you of who the players are. The countercurrent multiplier of the nephron loop depends on 2 things. Okay, so when we're talking about countercurrent multiplier, the structure, the anatomy, is the loop of Henle in juxtamedullary nephrons. The 2 factors that create this gradient is filtrate flow in opposite directions as well as the differences in permeability. The descending limb, okay, so we're going down and we're going to go back up. The descending limb is permeable to water. So water can leave through that paracellular or through aquaporins, and it can leave. When we get to the ascending limb, aquaporins are not present and the junctions are tight. So water is going to have to stay in the tubule. However, salt is going to be actively transported in that ascending limb to the interstitial fluid. So countercurrent and differences in permeability, if we don't have those, then we can't create that gradient or maintain it. The more salt the ascending limb actively transports out into the interstitial fluid, the more water diffuses out of the descending limb. Okay, difference is multiplied along the length of the loop. So this multiplied just means it is continually increased from the cortex medullary boundary down to the bottom of the loop. In order to understand how this gradient is created, it's important to start actually at the ascending loop. So even though our filtrate flow is, we have our glomerulus to the proximal convoluted tubule, and then it's going to enter the loop of Henle, we need to look at this and then descending, ascending, and then we're going to come up to the distal convoluted tubule down to the collecting duct. This is the DCT. In order to understand how this is going to work, we really need to look at this ascending limb first. In the ascending limb, water cannot move. It's stuck in here. But salt is actively transported out into the interstitial fluid. So the salt concentration is going to increase both between this loop and then also on this lateral side. As salt is actively transported, the concentration is going to increase. Once we understand that salt is continuously being pumped out, then we can look at the descending limb. With the descending limb is permeable to water. And so we're going to start here. We're about 300 milliosmol. And then we get this active transport on the ascending limb of salt. So that salt concentration increases. And because of osmosis, water is going to move. Now we're going to go and do a little primer on osmosis because it's been a little bit for some. So if we have a beaker and we have a semi-permeable membrane, which in this situation is going to be aquaporins. So aquaporins allow water to pass, but not salt. So if we are in the descending loop of Henle, and this is the interstitial fluid of the medulla, and then over here, so then we get our loop and it's going to come back over here, and this would be our ascending limb over here. Okay. The ascending limb is increasing the salt concentration. If we have high salt, that does not leave room for water. So a high salt part of the solution means that it is going to be low water. In the filtrate, we have in the descending loop, we have a low salt comparatively and a higher water. So if you don't have as much salt, then you have more room for water. And so that creates the movement of water through these aquaporins and into the interstitial space, interstitial fluid. So if you are shaky on osmosis, you may need to pull up a quick video to remind you of how osmosis works. So salt is being produced. Water says, oh, I need to go equilibrate. And osmosis is passive. There's no energy involved, but it does have to follow the concentration gradient. So as water moves out, you can look at the color of the filtrate as it moves down through this tubule system. Water is leaving, which means the salt concentration in the filtrate is increasing. As the salt concentration increases and increases because water keeps moving out, it gets very, very concentrated, down to 1,200 milliosmoles. And then as it comes out and is starting to go up the ascending limb, now we get the salt can pump out, but it also it's very concentrated. And so it keeps pumping out more and more salt. And then so the concentration of salt is going to decrease. So on the descending limb, we move water from the filtrate into the interstitial space, and then the vasorecta capillaries are going to take that water and reabsorb it into the systemic system. On the ascending limb, we get salt being removed. So going into the loop of Henle, we've already removed about 70%. In order for us to survive, like live like more than an hour or a couple hours, we need to remove 99 to 99.5% of both salt and water in order to maintain plasma volume. So 70% is just not going to cut it. So we need to reabsorb more. Water will be reabsorbed because it's allowed to move out of the descending limb. Salt is actively transported out into this interstitial fluid. The vasorecta captures that salt and returns it to the systemic system. So the loop of Henle has two functions: the reabsorption of water and sodium chloride back to the systemic system. That'll get us like almost 90% of where we need to be. The other second function is to create this gradient where we have 1,200 milliosmol at the bottom up to 300 osmol at the top. This gradient is not going to be why we reabsorb salt and water in the loop of Henle. These are kind of two separate events that happen at the same time. Okay, so we reabsorb water, we reabsorb salt, and we create this gradient that will then be used by the collecting duct. So another picture, we get afferent arterial, glomerular capsullaries, efferent slightly smaller, creating hydrostatic pressure. Hydrostatic pressure forces filtrate out. The osmolarity of the filtrate is the same as the plasma because it is plasma, just minus plasma proteins. It goes to the proximal convoluted tubule where we reabsorb 100% of glucose, 100% of amino acids, and then about 65 to 75% of water and sodium. We start going down these descending limb. Water can move, salt cannot. So water is going to move, and it is assisted by this gradient, but that's not the main function of this gradient. So the water is going to move into the interstitial fluid. It will be captured by the vasa recta and returned to the systemic circulation. Then when we get to this bottom loop, we're at peak osmolarity, because as the water moves, the salt cannot, and so it gets more and more concentrated. As the filtrate moves up the ascending limb, heading towards the distal convoluted tubule, salt is actively transported out. And it's a pretty consistent rate. And if you notice, there's a difference of 200 milliosmoles at every level between the descending limb and the ascending limb. So much salt is pumped out that by the time it reaches the distal convoluted tubule, it's what is called hypotonic. There's a lot less solute in the filtrate that is leaving as compared to when it entered. But the volume has also been greatly reduced. So we've reduced volume over here. We've reduced solute concentration on the ascending limb, so that when it hits the DCT, it is a much smaller volume and it is hypoosmotic. So it's at 100 milliosmoles. So right now there's not a whole lot of urine, but it is still very dilute. I'm just going to briefly talk about the vasa recta. We're not going to go into the numbers and the mechanism as much. You just it's one of these things you're going to kind of have to trust me, it works. The countercurrent exchanger utilizes the vasa recta. The vasa recta is called countercurrent because the vasa recta, this is the vasa recta. Okay, this is not the tubule. It's not my favorite picture, but it's really not a better one. So this is the vasa recta, as you can see. And if you remember from lab, it overlies that loop of Henle. So it's like a mesh of capillaries that travels the same pattern as that loop of Henle. So you'll just have to draw on that for kind of a visual reference. The vasa recta is going to capture the water and retain the salt as it returns to the venous system. And because its countercurrent pattern is the same as the loop of Henle, it doesn't change the osmotic gradient in the medulla. So it removes reabsorbed water, and it will reabsorb some salt, but it does not, it doesn't do it in a rapid way to disturb the gradient. Okay. So the gradient is preserved, but salt and water to some extent are going to be, definitely water and salt to some extent will be reabsorbed into the systemic circulation. So the countercurrent exchange is accomplished by the vasa recta. It will return reabsorbed water to the systemic system along with a small amount of salt. Okay, so we've now established that gradient where we have low salt to high salt. And at this loop, we can't get this high of osmolarity using salt alone. And so urea is going to contribute to the medullary gradient. To get between 900 and 1200, you need to increase the contribution of solutes beyond just that of salt. And urea is going to be that solute that does that. We're going to establish, so we've established the osmotic gradient, and that can now be used to form dilute or concentrated urine. Without the gradient, we would not be able to raise urine concentration above 300 milliosmol. If you're overhydrated, like you've been chugging water or having like lots and lots of lemonade, then the urine concentration will remain about 100 milliosmoles. So what it's coming out at here, it just continues down the collecting duct and then out. We'll talk about the pathway, ureters, bladder, to be excreted. If we are suffering, wrong direction. If we are suffering from significant dehydration, then aldosterone can be secreted, and that will allow for additional ions and then water to move to further dilute the urine. But most of the time, I think I might have said extremely dehydrated, extremely overhydrated, then we can make it even less dilute. So if normal, like been drinking a lot, we'll stay at 100. But if we really need, this usually comes more into play with blood pressure also, we can further reduce the osmolarity. Oops. For dehydration, so dehydration is going to cause ADH to be secreted from the posterior pituitary, and ADH is going to influence the number of aquaporins in that collecting duct. If someone is extremely dehydrated, urine can be concentrated up to 1200 milliosmol. That would be pretty unusual. Usually even really concentrated urine is more around 900. So that would be under extreme conditions. So this is showing someone that is well hydrated. ADH is not going to be secreted, or it will be secreted in very low levels. The filtrate that comes out is 100 milliosmoles, and it will continue to be a large volume of dilute urine that is excreted from the body. If somebody is like most of us, we don't drink enough water, then we our body realizes that we are dehydrated. Depending on how dehydrated we are, the posterior pituitary secretes antidiuretic hormone. Antidiuretic hormone is going to move aquaporins into the membrane of that collecting duct. And when aquaporins are placed in the collecting duct, then water is going to move. And depending on how dehydrated we are, will depend on how many aquaporins are present and where in the collecting duct they are. So if we just need to concentrate a little bit, we put aquaporins in here in a moderate number. If we need to reabsorb like most of the water, like nearly all of it, then we'll put them in through the entire range of the collecting duct. And this reabsorbed water is also going to be transported by the vasa recta into the systemic system. As I mentioned, and this is about all I'm going to mention it for now, is that we have nitrogenous waste. Okay, I thought I had another one. Okay, so we have nitrogenous waste. So we get urea from amino acid breakdown. We'll talk about creatinine from skeletal muscle metabolism. So there are nitrogenous wastes that need to be removed. The body will also use urea to alter the osmolarity gradient in the medulla. So at the very hairpin part of the loop, urea is secreted into the interstitial fluid to increase the osmotic level in that medulla. So that's really all I'm going to say about urea, is that it contributes to that medullary gradient. There are many chemicals, substances that can enhance urine output. Alcohol is a big one. It inhibits ADH. So without ADH, then aquaporins are not placed into the membrane and water is not reabsorbed. So alcohol, if you have noticed, when people go out drinking, they seem to have to go like to the restroom like every hour. And that's because the urine is very dilute because we're not reabsorbing water. Caffeine is going to it's going to affect sodium reabsorption, and as a result, water reabsorption. It also results in dehydration. The dehydration effects of alcohol and caffeine are what lead to headaches and hangovers. Loop diuretics, they inhibit medullary gradient formation. They are going to affect salt and forming the gradient, and then other diuretics. We're going to move on to looking at renal clearance and then look at a few disorders. Renal clearance is the volume of plasma that kidneys can clear of a particular substance in a given time. Renal clearance tests are used to determine glomerular filtration rate. So healthy kidneys are going to produce, for 2 kidneys, about 125 mils per minute. So for 2 healthy kidneys, that is the glomerular filtration rate. If you put a substance into the blood, the first one we're going to talk about is inulin. Inulin is not produced by our bodies. It's a plant polysaccharide, but it is filtered at 100%. So it is not secreted, it's not reabsorbed. We put it into the blood, it goes into the filtrate, it goes through the tubule system, and then we measure it in the urine. If kidneys are functioning well and as expected, the clearance of inulin would be equivalent to glomerular filtration rate. So it would equal 125. If it is lower, then a substance is being reabsorbed or not filtered properly. And then if it's over, things are secreted. Some substances have a renal clearance of 0, and that renal clearance of 0 means that they are usually not filtered. So glucose and amino acids should have a clearance of 0 because they should be completely reabsorbed and they shouldn't be in the urine. Okay, so inulin is ideal because it is freely filtered, not reabsorbed, not secreted, but you have to infuse it via IV, and that's just not, that's a huge hassle. So instead, we use creatinine clearance as a measure to estimate GFR. So creatinine has a clearance value of 140 mils per minute. It is freely filtered, meaning if it's in the plasma, it ends up in the filtrate in the same concentration. But there is also a small amount that's secreted into the proximal convoluted tubule, so slightly over. However, in clinical terms, we just call it good, that if creatinine clearance is 125 or so, then glomerular filtration rate is good and kidneys are healthy. If creatinine clearance is shown to be lower and used as an estimate of GFR, then it can be an indicator of renal disease. For the exam, you are not going to have to calculate renal clearance rate. You do need to understand that renal clearance is an estimator for glomerular filtration rate, and it will follow the progress of renal disease. Okay, so we use it to estimate how well our kidneys are functioning. In chronic renal disease, it's defined as a glomerular filtration rate, which we usually measure as creatinine clearance, of less than 60 mils per minute for a period of 3 months. Filtrate formation decreases, nitrogenous waste accumulate in the blood, and the pH becomes acidic. We'll talk about how this is compensated for next week. It can be seen in diabetes and hypertension. Also, sometimes exposure to different toxic chemicals can damage kidneys as well. Renal failure is defined as a glomerular filtration rate less than 15 mils per minute. This is when a patient's probably going to be put on dialysis well before this, so somewhere between here and here, because you don't want a kidney to get to failure. Okay, so now we're going to talk about actually getting rid of urine. We talked about, so we have the distal convoluted tubule and the collecting duct are under hormonal control to determine if urine is going to be dilute or concentrated, and then it needs to get transported and excreted. Urine should be clear, and it should, cloudy indicates usually an infection. It should be pale to deep yellow depending on hydration status. Urine is, in a Western diet, tends to be acidic because we tend to like acidic foods. If somebody is vegetarian, vegetables contain alkaloids. And so if somebody is truly like vegetarian or vegan, their pH may be more on the alkaline side. Specific gravity looks at the density of urine in comparison to the density of water. Water has a specific gravity of 1. So if someone is very well hydrated, the urine would be dilute, meaning not as having as many solutes, and would be around 1.01 to 1.02. As a person becomes more dehydrated, then it becomes more dense and the specific activity would increase. So you would see a specific activity of someone who is dehydrated to be more around 1.03. So specific gravity increases as urine becomes more concentrated and is a reflection of hydration status. Urine is usually about 95% water, 5% solutes. It does contain nitrogenous waste. Urea from amino acid breakdown is the largest component, then smaller contributions from uric acid and creatinine. There are also other ions that are found: sodium, potassium, phosphate, sulfate. Calcium is going to be hormone dependent. Magnesium and then bicarbonate is going to be reflective of acid-base status, which we will talk about next week. Abnormally high concentrations of any part can indicate pathology. This table is kind of interesting. So normally glucose should be reabsorbed 100% at that proximal convoluted tubule. But because it uses facilitated diffusion, it uses a transport protein, there is that maximal transport rate. If the amount of glucose going in to the filtrate exceeds the Tmax, it will then get excreted into the urine. This occurs in uncontrolled diabetes. So for proteins should not be filtered. So amino acids get through, but proteins should be retained in the glomerular capillaries. If proteins are not contained, then they will get into the filtrate, not get reabsorbed, and get excreted in the urine. Sometimes that just happens due to non-pathological excessive exertion. Pregnancy can indicate an underlying pathology. So one of the things that is monitored during pregnancy is protein in the urine, because it can be associated with preeclampsia, so hypertension involved with pregnancy. And sometimes the women are very borderline on hypertension, but they can still be preeclamptic, which it can have serious delivery implications. So proteins in the urine could be due to, there's a problem in the filtration membrane, there's a hypertension, preeclampsia, glomerular nephritis. So somebody gets a severe kidney infection, it inflames the that membrane, proteins can start going through, often an initial sign of renal disease. Ketone bodies, also usually associated with diabetes, hemoglobin, sometimes hemolytic anemias, bile liver disease, erythrocytes are bleeding in the urinary tract due to trauma, kidney stones, infection, or cancer. One of the interesting things about stomach cancer is that it will show up as blood in the urine. And it's one of the few indicators of early disease for stomach cancer. Leukocytes, so white blood cells, urinary tract infection. So you might see a few of these in some sort of form of a question on the exam as to possible cause and what's happening. So we've gone through that whole process of forming filtrate and then reabsorbing water and reabsorbing salt and reabsorbing glucose and reabsorbing amino acids. And we finally get to the collecting duct where we get urine at the end at the patella, and it's dumped into the calyces to the renal pelvis, and then ending in the ureters. So both the ureters and the bladder are going to have 3 layers. The first layer is made up of a transitional epithelium, so it can change shape depending on fluid presence. Then a muscular layer, and in the ureters, so this is that transitional epithelium, and then in the ureters you have longitudinal and then circular, and then this adventitia connective tissue. The arrangement in the bladder is slightly different, but still 3 layers: transitional epithelium, smooth muscle, and adventitia. I always like to call out this, smooth muscle layer, because urine does not move by gravity alone. And that should be intuitive, and that we go to bed and we are horizontal. And what is the first thing we need to do when we wake up? We need to go to the bathroom. Okay, so that means urine has been formed and propelled through the evening as we're sleeping. So it is not a passive process. It does involve smooth muscular propulsion. As ureters propel urine down to the bladder is a muscular sac for the temporary storage of urine. It doesn't sound very exciting, but it actually has quite a bit of nervous innervation. So we'll talk about that. The normal holding capacity is 500 mLs, but your initial notification to your brain that you may want to start emptying your bladder is usually closer to 250 mLs. The bladder with enough sympathetic nervous stimulation can hold more than 500 mils, going up to 800 or 1,000. Again, there are 3 layers: mucosa, and then the muscular layer, but it's now reversed with the circular in the middle and the longitudinal outside. Not that you need to remember that right now, or maybe ever, but you do need to know that there are in this fibrous connective tissue, that the same 3 layers of mucosa, transitional epithelium, smooth muscle, and the fibrous connective tissue are the same in the ureter and the bladder. You've got ureters that drain into the bladder. The bladder is going to store urine, and then urine release will be controlled by an internal urethral sphincter and an external urethral sphincter. And then it will, as it passes through the urethra. We'll talk more about the urethra when we get to the reproductive system. It's very short in women and much longer in men. Like I said, usually by this time we're running out of time. We're getting close. So we save the anatomy and the tissue structure for the reproductive system. The internal urethral sphincter is under autonomic nervous control. The external urethral sphincter is under voluntary, so skeletal muscular control. It is considered a reflex. Okay. So there are 2 sphincters that control urinary bladder drainage. The internal one is controlled by the autonomic nervous system. It is under involuntary control. It receives input from both the parasympathetic system and the sympathetic division of the autonomic nervous system. The external urethral sphincter is under voluntary skeletal muscle control. So micturition is also called urination, and 3 events must occur. The contraction of the detrusor is the main muscular layer in the bladder. The internal urethral sphincter opens, and then the external also opens. This is a better, actually I'm gonna go to this one first. So at rest, the bladder is relaxed. There are stretch receptors in the bladder. And so they are not being activated. So they are not sending messages to the integration, the pons, we'll talk about the pontine centers, to initiate this reflex. However, we are getting tone, voluntary muscle tone, to keep this external sphincter closed. But it doesn't require a lot of effort because this one is closed as well. Then as the bladder fills, so the stimulus is an increase in bladder volume. That stimulus then is going to be received by stretch receptors in the bladder. When these stretch receptors sense an increase in volume, so they're mechanoreceptors, they get stretched, increase in action potentials, that will be sent to these 2 centers, which are on the previous slide, activating the parasympathetic system, inactivating the sympathetic division. It's I can't find a diagram I really like. The first one's too busy. This one doesn't include the sympathetic. And then also stimulating the motor neuron to open the external sphincter. Again, the stimulus is urinary bladder filling. It is stretching the walls. It is going to stimulate stretch receptors in the bladder. And then there are kind of two, so it'll notify the higher brain center. And this first starts at 250 mils. The brain, you're going to go, it's a good time or it's not a good time. I think I'm going to wait till after lecture. That's a good idea. Or no, I'm just going to go ahead and go now. If if your brain decides that it is not a good time, then there's a nuclei in the pons that then will send messages to decrease parasympathetic activity, increase sympathetic activity, and then increase somatic motor nerve activity. These two will keep those sphincters closed. So the sympathetic says, no, we're going to keep the internal sphincter closed, and somatic says we're going to keep the external sphincter closed. So both of those are going to stay closed. And how you can think about this is sympathetic activity is fight or flight. And, we get scared, we're running from the bear, sympathetic nervous activity increases. The last thing we want to think about is trying to find a place to go to the bathroom. So increased sympathetic activity is going to inhibit micturition or urination. If it is, you decide that it's a good time, then there is a separate set of neurons in the pons that is going to increase parasympathetic activity, which releases or relaxes that internal urethral sphincter. It will also inhibit sympathetic activity. And then now there's also the decision that yes, we're going to go to the bathroom. So sympathetic motor nerve activity decreases, allowing the external sphincter to open. So we have an open internal sphincter because of this increase in parasympathetic, and opening the external sphincter because of decrease in motor activity. And then the bladder will contract and urination occurs. Seems very complicated for something that we do many times per day. The things for the exam to keep in mind are the stimulus, which is bladder filling, the receptors, which are stretch receptors in the bladder wall, and that parasympathetic activity encourages micturition, whereas sympathetic activity inhibits it. So just the general results of activation of the sympathetic nervous system, general results of activation of the parasympathetic system, and then knowing that the autonomic nervous system controls the internal urethral sphincter, the voluntary motor system controls the external one. So more big pictures, not terribly in detail on this one, just like I said, big picture for this reflex. The big details are in glomerular filtration and what's reabsorbed where. Okay, so those we've spent an entire lecture on. There will be finer details in those sections. This is just, the other thing I was gonna say is, so you get, like, your bladder starts filling, and about 250, it'll notify your brain. Your brain decides, I'm good, I'm gonna wait. It takes a while for that reflex to respond again. So you've got about another 50 mils, which is for most individuals, depending on hydration status, would probably be about 40 minutes until it sends another signal to your brain saying, okay, So anyway, I just find that interesting. This is just what you would find in the urine per day. A lot of urea, should be 0 glucose, should be very little protein, and then this is usually around the amount of urine produced per day. This was to remind you that I think it would be helpful to draw the blood vessels and to draw the nephron loop system, identify the segments, identify what is reabsorbed where. You don't need to go into this great a detail, but I just grabbed this off of a Google search because I was like, oh yeah, just remind me to remind them. Then I would also add like that water can leave the descending, sodium can leave the ascending, things like that. But with kidneys and nephrons, I find drawing is maybe therapeutic, but also very helpful as a study guide. Okay. That is the end of chapter 24.
The permeability of the principal cells to sodium ions in water is controlled by 2 hormones, aldosterone from the adrenal gland and antidiuretic hormone, or ADH, from the posterior pituitary gland. Let's first look at the role of aldosterone, which precisely regulates the final amount of sodium reabsorbed. When levels of sodium and potassium ions in the blood are balanced, aldosterone levels remain low. As a result, there are few sodium-potassium ATPase ion pumps in the basolateral membrane and few sodium and potassium channels in the luminal membrane. Therefore, sodium ion reabsorption and potassium ion secretion are both low. However, a decrease in the level of sodium ions or an increase in potassium ions in the blood will trigger the release of aldosterone. Let's see the results of increasing the level of aldosterone in these cells. In response to increased aldosterone, both sodium ion reabsorption and potassium ion secretion increase. This occurs because the principal cells increase the number and activity of sodium-potassium pumps in the basolateral membrane. The number of sodium and potassium channels in the luminal membrane is also increased. Notice the absence of potassium channels in the basolateral membrane. Potassium ions enter the cell through the basolateral membrane, but instead of diffusing back to the interstitium, they diffuse to the luminal membrane and are secreted into the filtrate. The effects of aldosterone are not confined to the collecting duct. It has similar effects on cells of the distal convoluted tubule. You should also notice the resulting increase in interstitial osmolarity. Water is not following the solute because the luminal membrane of the collecting duct is relatively impermeable to water unless it is stimulated by ADH. Aldosterone and antidiuretic hormone act independently to regulate salt and water, but can also work together. The cell you see here has been stimulated as yet only by aldosterone, so it is still impermeable to water. Let's see the effect of the pituitary gland releasing ADH. When stimulated by ADH, principal cells quickly insert luminal water channels, increasing their water permeability. Notice that the interstitial osmolarity decreases. When water molecules can diffuse through a membrane, osmolarities on each side of the membrane equilibrate. Using this schematic diagram, let's review how filtrate concentration in the tubules is related to interstitial osmolarity. Watch the changes in the concentration and volume of the filtrate as it passes through the differing osmotic environments of the cortex and medulla. Since the cells of the proximal convoluted tubule, or PCT, are highly permeable to both solutes and water. the relative osmolarity of the filtrate remains equal to the 300 milliosmol solute concentration of the interstitium. The cell's high permeability also accounts for a 65% reduction in filtrate volume. Watch the simulated drop of filtrate as it moves down the descending loop of Henle to the bottom. Notice that the osmolarity of the filtrate increases and the volume decreases. Recall that the cells of this region are permeable to water but not to solute. As the filtrate moves down the tube through regions of higher osmolarity, water diffuses out into the interstitium, reducing the filtrate volume by an additional 15%. The solutes remain behind in the tubule and become more concentrated as the filtrate approaches the bottom of the loop. The cells of the thick segment of the ascending loop of Henle are permeable to solute, but not to water, making them function essentially opposite to the cells of the thin segment of the descending loop. As the concentrated filtrate flows up the ascending loop, the cells actively transport solutes into the interstitium, causing the osmolarity of the filtrate to fall to less than 300 milliosmoles. Because water remains in the tubule, the filtrate volume remains unchanged. The opposing flow and opposite activities of the descending and ascending segments of the loop of Henle is called the counter-current multiplier mechanism. The osmolarity of the filtrate entering the late distal convoluted tubule, or DCT, and cortical collecting duct can be as low as 100 milliosmoles. Recall that in the cells of this region, the reabsorption of sodium ions and water is regulated by the hormones aldosterone and antidiuretic hormone. In normal hydration conditions, low levels of both hormones promote the reabsorption of sodium ions and water from the filtrate. This maintains the low osmolarity of the filtrate while reducing its volume by an additional 15%. The osmotic gradient constructed by the countercurrent multiplier mechanism concentrates the urine by drawing water from the filtrate as it travels through the medullary collecting duct. However, the degree of concentration is regulated by antidiuretic hormone, which controls the water permeability of the duct. As seen earlier in this topic, ADH levels vary in response to various conditions, including the individual's hydration status. Let's observe the normal level of hydration first, and then compare that condition with the other two hydration levels. Notice the effect of each condition on the drop of filtrate traveling down the medullary collecting duct. With normal hydration and levels of ADH, water channels are present in the luminal membranes of these cells, resulting in moderate water permeability. ADH also facilitates the diffusion of urea out of the medullary collecting duct into the interstitium. Although it is considered a nitrogenous waste product, urea is responsible for up to 40% of the medullary interstitial osmolarity. From the interstitium, urea passively re-enters the filtrate in the loop of Henle and recirculates back to the collecting ducts. It may then again diffuse into the interstitium or pass into the renal pelvis as a component of urine. Notice that as it descends, the filtrate drop shrinks in volume and darkens slightly as water is lost and solutes are concentrated. The filtrate does not equilibrate with the osmolarity of all medullary regions and is therefore not as concentrated as possible. Normal urine has an osmolarity of about 600 milliosmoles, or twice normal body osmolarity. With dehydration, a high level of ADH creates two important changes. First, it causes additional luminal water channels to be added to the duct, which increases its permeability to water. Second, it increases the permeability of the duct to urea, which in turn increases the interstitial osmolarity. This increased osmolarity draws additional water from the filtrate. Therefore, as the filtrate passes through the lumen of the duct, it equilibrates with each regional increase in osmolarity. Notice the decrease in size and darkening color of the filtrate drop as it descends through the duct. In severe dehydration conditions, the low volume of urine excreted may be concentrated to about 1,400 milliosmoles, or more than 4 times the osmolarity of normal body fluids. With overhydration, ADH levels are very low or absent, and the duct cells remain relatively impermeable to water and urea. The reduction in urea permeability decreases the medullary interstitial osmotic gradient, reducing the water-drawing power of the interstitium. As the filtrate passes through the lumen of the medullary collecting duct, it does not equilibrate with any regional change in osmolarity and therefore remains unmodified. Notice that the filtrate drop remains the same size and color as it descends through the duct. The final urine, which is dilute and high in volume, may have an osmolarity as low as 100 milliosmoles.